morning to all of you. Today we are going to study something detailed about the pollen grains, or it is known as the microsporangium. So it is the part of the chapter number 2, sexual reproduction in plants. That is the part 2. So pollen grain or microspores. So what is actually pollen grain or microspores? Pollen grains representing the female gametophyte. Actually, female gametophyte is forming from the pollen grains. So pollen grains are representing the immature male gametophyte, not female, male gametophyte. So pollen grains are representing the male gametophyte, the immature male gametophyte. This pollen grains is produced in the anther. So already we studied the microsporangium, the structure of the microsporangium. In the microsporangium there are four, in the anther there are 4 microsporangium, and the microsporangium containing the sporogenous tissue, and the sporogenous tissue undergoing meiosis and producing the tetrad, and this is the tetrad. The tetrad having 4 pollen grain in the immature pollen grain. When the anther undergoing the dehydration and also this structure undergoing the dehydration, this tetrad will separate from each other and forming the 4 viable pollen grains. So this is the tetrad. The tetrad will lose and it will become the pollen grains. So that is the pollen grains. So pollen grains representing male gametophyte. Male gametophyte. So this is actually immature male gametophyte, not a mature male gametophyte. From this pollen grain, mature gametophyte will develop. So what is the structure of the pollen grains? So we are going to study the structure of the pollen grains. A pollen grain is spherical in structure, spherical, golakar, or it is near about spherical in structure. Most of the flowering plants, it is spherical in structure. It is about near about 25 to 50 micrometer is in the diameter of the pollen grains, and each pollen grains having 2 layers. It is 2 covers, 2 layers. So what are the 2 layers? One layer present inside and another layer present outside. This is the outer layer and one layer present inside. This outer layer is known as the exine, E-X-I-N-E, exine, and the inner layer is known as the intine. So now what are the differences between exine and intine? First let us see the exine. The exine is made up of a particular chemical. The name of the chemical is sporopollenin. Sporopollenin. This sporopollenin is produced by the 4th layer of the microsporangium, that is the tapetum. Tapetum producing the sporopollenin, and the sporopollenin forming a thick layer outside the pollen grains. That thick layer is known as the exine. And this sporopollenin is very resistant to the high temperature. Low temperature, no problems. It is high temperature and also high concentration of the acid and highly concentrated alkali. The sporopollenin is very resistant, and the sporopollenin is also resistant to the ever-known enzyme. There is no enzyme still we know that the sporopollenin can be digested. So such a very strong material is the sporopollenin. This is the most or the strongest material that we know until today. So the sporopollenin forming exine, and this exine is not entire. This exine showing some openings, some pores in between. Intermittently it is showing some spores. This pores is known as the germ pore. So this is the germ pore. Germ pore. Why it is called a germ pore? Because the germination of the pollen grains is taken place through this germ pore. So during the germination, the intine will come outside as a tube-like structure that is known as a pollen tube, and this tube is coming outside through this pore. So it is known as a germ pore. And as a result, the sporopollenin is very strong. So it can be preserved in the ice or in such conditions. So when we digging the ice or digging the mine, we can obtain the sporopollenin. We already obtained many pollen grains, and this preservation, high preservative properties because the presence of the sporopollenin. Okay, so that is the outer cover, and then coming the inner layer. The inner layer is known as the intine. The intine is made up of the 2 chemicals, that is the cellulose and pectin. These are the 2 two chemicals, polysaccharides, cellulose and pectin. So intine is made up of the cellulose and pectin. It is present inside and it is entire. It has no opening. So during the germination, this intine is coming outside through the germ pore as a tube-like structure. Okay. So inside the intine, so this is the intine. Inside the intine, there is a thin layer that is known as the plasma membrane. So it is a cell. So plasma membrane is common to all the cell. So plasma membrane present inside the plasma membrane, cytoplasm present. This is the cytoplasm. In the cytoplasm, there are two cells. Actually, one at the beginning only one nucleus present. So this is the one nucleus, and this nucleus will divide, and one nucleus remaining upper side, one nucleus remaining lower side, and the cytoplasm, that is the karyokinesis complete. After the karyokinesis, actually the mitosis, after the karyokinesis, it divides unequally, and one cell present lower and another present in the upper side. This is the largest, one is the larger cell and one is the smaller cell. See here, this is the nucleus, and the nucleus, this is the vacuole. It's a plant cell, so it is containing the vacuole. This is the nucleus. The nucleus is going to divide, and here the spindle fiber forming, and this spindle fiber forming asymmetrically. If it's symmetrical, both the cells will cell formed after the mitosis, both the cell will be equal. So asymmetrical spindle fiber formed. So one cell become larger and one cell become smaller, and it producing the two cell. Out of the two cell, one cell, the large cell, it is known as the generative cell. So this is the tube cell, or it is called a vegetative cell. So large cell, that is called a vegetative cell, and a smaller cell, that is known as the generative cell. It is a vegetative cell because it is a one point, it is a larger one comparing to the another one, and it has the nucleus. This nucleus is not spherical, it is irregular shaped nucleus. There is another identification of the vegetative cell, and it is called vegetative because it stores the food materials. The food materials requiring for the growth or the maturation of the pollen grains. Food is requiring. This food is stored in the vegetative cell. This vegetative cell is also known as the tube cell. It is also known as tube cell. So that is the vegetative cell. And the generative cell, it is a spindle shaped. Spindle shape means both ends pointed, so it is known as spindles, and the nucleus is present inside. It is containing abundant cytoplasm. So this containing abundant cytoplasm, and this containing abundant reserved food materials. So it is spindle shaped. Another very important point, actually the spindle shaped generative cell, it has no cell wall, and that freely floating inside the vegetative cell. So vegetative cell has the cytoplasm. In the cytoplasm of the vegetative cell, this generative cell freely floating. It is spindle shaped, it is smaller and floating in the cytoplasm of the vegetative cell, because a cell wall is absent in the case of the generative cell. So in most of the means near about 60% plants, 60% flowering plants, the pollen grains is liberating at this two-celled stage. So this is the two-celled stage. So 60% flowering plant, the pollen grains are releasing two-celled stage, and the remaining 40% plant, flowering plant, the pollen grains are releasing at a three-celled stage. So which one is the third cell? In some of the pollen grains, the vegetative cell. The vegetative cell remaining there, this is the vegetative cell, and this is the generative cell. This generative cell divides and producing the two male gamete. So this is the one cell, this is another cell, and this one is the vegetative cell is the third cell. So in the third three-celled stage, 40% flowering plant, three-celled stage, the pollen grains are liberating. Okay. So that is the liberation. So when the pollen grains is liberating, or the pollen grains liberating at a two-celled stage or three-celled stage, such questions are asked in the NEET examination previously. Or it may be three-celled stage or it may be two-celled stage. But the two-celled stage is the more predominant, because 60% plant two-celled stage the pollen grains will release, and only 40% plant the three-celled stage the pollen grains will release. So that is the structure of the pollen grains. So this is the generative cell, this is the vegetative cell, it is also known as the tube cell, and here is the asymmetrical spindle formation as a result of two different sized or one large and one small cell will produce as a result of this asymmetrical spindle fiber. Okay, so that is the structure of the pollen grains. Then say what is the significance, economic importance of the pollen grains. Pollen grains having economic importance. So what are the economic importance of the pollen grains? Economic importance of pollen grains. One of the economic important, it is helping for the pollination. After the pollination, fertilization taken place and seed will form. Seed and fruit will form. That is one of the economic important. That is related to the plant. Another economic important, particularly related to the human welfare. These pollen grains are very dangerous because it is causing some of the allergic disease. So the most of the pollen grains causing allergic disease like asthma disease or respiratory problems. It is causing respiratory problems. So it is microscopic, and when it enter through the nose and the throat, it remaining, or it may be enter to the trachea and then to the lungs, and that is causing the allergic disease. And for a very good example for that, that is the Parthenium. There is a plant Parthenium. Parthenium is called carrot grass. Carrot grass. So there is a congress grass also mentioning the Parthenium. The Parthenium is not the indigenous. It is not present. It was not present in India. It import along with the wheat. The seeds are imported in India. Nowadays, the Parthenium is a very headache to the government, just like terror of Bengal. Water has in the water. It is a very headache to the government. Like that, Parthenium cannot be eradicated in such a fashion. It spread on each and every nook and corner of the earth. So this Parthenium producing when the flowering, the Parthenium, the flower is opened and the pollen grains will spread, and this pollen grains are causing the respiratory very acute and even the chronic respiratory symptoms are causing by this Parthenium carrot. See, another this is the pathogen, this is a bad effect, and one of the good effect, it is containing the nutrition. The pollen grains are containing nutritive. It is very nutritious, very important nutrients, vitamins and other minerals that are present in the pollen grains, and these pollen grains are collected and made paste, and then it prepared the tablets, and these tablets are available in the market. So that is a very nutritious, and these tablets also, not only tablets, it also from the pollen grains also prepared some syrup and a tablet and other product, and this product is a very healthy product, and the athletes and even for the horse, the stamina, increasing the stamina or the performance of the athletes, and for the fast running or the performance of the horse, this tablet and syrup is using syrup and using for the athletes, and even for the horse, it is giving to the horse for the better performance. So that is the economic importance of these pollen grains or the microspores. Then see, these microspores or the pollen grains, when shedding at a 2-celled stage or at a 3-celled stage, it is releasing from the anther. It is shedding from the anther. After shedding from the anther, it is landing on the stigma. So this is the female reproductive structure. It is called a carpel. So this is the ovary. This is the thalamus. This is the ovary, and this is the so this is ovary, this is the style, and this is the stigma. Okay, so this pollen grains it is falling on the stigma. After falling on the stigma, compatible pollen grains, when falling on the stigma, it is germinated and then produce the pollen tube, containing the gamete. That is the male gametophyte. So here, what is the point? The pollen grains must fall on the stigma before losing the viability. So the viability means the healthy condition. The pollen grains must falling on the viable condition or in the healthy condition, otherwise it cannot germinate. So this viability period is different from one plant to another plant, one species to another species. Viability means full strength. So before losing the strength, it must be falling on the. After once the viability is lost, it cannot germinate on the stigma. So this viability is depending upon the two conditions, that is the temperature and humidity. Temperature and humidity. These are the two conditions that are requiring for the maintaining the viability. Humidity. So if suitable temperature and humidity is there, these pollen grains will remain viable for a particular period. Then during this period, when it is falling on the stigma, it will germinate. And this viability period is different in different species. For example, in the case of the rice plant, rice and wheat. The rice and wheat, it is a 30 minute. So only 30 minutes. Within 30 minutes, it must falling on the stigma. Then only it germinate. Otherwise, it cannot germinate. But in some other plant, for example, Rosaceae, the family Rosaceae, the family Rosaceae, the rose plant belong to the Rosaceae family. Another one, Leguminosae, pea plant, Leguminosae, and Solanaceae, potato family, Solanaceae. In this family, the pollen grains can remaining for 1 month. So it is remaining for 1 month. So within 1 month, the pollen grains must fall on the stigma. Then only it is germinated and the male gametophyte, mature male gametophyte will produce. So that is about the viability of the pollen grains. Viability is the period. In that period, the pollen grains will remain in a healthy condition, and this during this viability period only the pollen grains can germinate. It is different in different plants. Okay, these pollen grains can be, so viability is a very short, 1 month or 30 minutes like viability. So nowadays the viability can be increased by the preservation. It can be preserved at a low temperature. Preserve at a low temperature. Low temperature means liquid nitrogen. If using the liquid nitrogen, we can lower the temperature minus 196 degrees centigrade. In minus 196 degrees centigrade, the pollen grains can be stored for many years. So in this low temperature, the low temperature can be maintained by using the liquid nitrogen. Under the low temperature, minus 196 degrees centigrade, it can be stored for long time. That is storing place at the low temperature is known as the pollen bank. So nowadays pollen bank is available. Previously gene bank, now also gene bank, and nowadays seed bank also available. Seed can be preserved at a low temperature. Like that, pollen also can be stored, preserved at a low temperature, and whenever it is requiring, we can remove from the low temperature, and then putting under the suitable condition, these pollen grains can be used for the artificial fertilization, hybridization process. So that is the pollen grains, the structure and other various aspects of the pollen grains. So okay, this lecture it is okay, and the next lecture I will explain the part third. I will explain the female gametophyte, female sex organs, gynoecium, and other different parts of the plant. So okay, thank you very much.